Hello and welcome to the next lecture of sexual reproduction in plants. In this lecture, we will discuss fertilization and the structure of seed. So let's start our discussion. Fertilization. What do you mean by fertilization? It is a process that involves the fusion of male and female gamete nuclei, or we can say sex cells nuclei. Suppose we have a male gamete and the female gamete. When these gametes meet, their nuclei, this is the male gamete nuclei and this is female gamete nuclei. During the process of fertilization, these nuclei fuse. And this process is called fertilization. Due to fertilization, a new cell forms that is called zygote. It is a single cell. So we can say the process of fusion of male and female gamete nuclei is fertilization. Nuclear term is very important. If you miss the nuclear term in the definition, the fertilization definition will become wrong. You cannot write process of fertilization is fusion of male and female gamete. No, it's wrong. Nuclear term is very much important because fusion of nuclei occurs. Okay, so in case of plants, male gamete is pollen grain, and pollen grain nuclei we can say is pollen nucleus. Female gamete is egg cell and we can say it has egg nuclei. So in case of plants in fertilization, when the pollen nucleus fuses with egg nucleus, the process of fertilization gets complete and it forms zygote. Zygote then divides many times. The cells of the zygote divides many times to form multiple cells. It divides single time to form 2 cells. Then it again divides horizontally to form 4 cells. And the process of cell division continues to form a bunch of cells. It is called embryo. This embryo further develops into a new individual. So this is the complete process of fertilization. Now one more thing to remember here is the zygote divides into multiple cells of embryo by the process of mitosis. Mitosis is type of a cell division. And another thing is male gamete is always small than female gamete. Female gamete is large. In case of plants and in case of humans as well. Next is, if you remember, we have discussed in the pollination that when pollination occurs, the male gamete lands on the stigma of the carpel. Stigma is the landing pad for the pollen grains. So when pollination occurs, pollen grain lands on the stigma of the carpel. Now pollen grain is here on the stigma and the egg nuclei is inside the ovule in the ovary. Then how will the fertilization will occur? How the pollen grain will reach to the ovule or the egg cell or egg nuclei? So it occurs by the process of formation of pollen tube. What happens when pollen grain lands on the stigma? Pollen grain absorbs some liquid from the stigma. Its enzymes, the enzymes of the pollen grains, become active and it starts germinating. The germination of pollen grain occurs and pollen tube start forming from the pollen grain. That is, pollen grain start forming pollen tube, and pollen tube, this is the pollen tube, the green one, it travels down the length of the style to reach to the ovary. Suppose this is the carpel. This is a stigma. This is a style. And this is ovary. Ovary has ovules and ovule has egg cell in it. Pollen grain will start forming a pollen tube and it will travel down the style to reach to the egg cell. Pollen tube grow down the style and reach to the ovary. In the pathway, it absorbs the cells of the style to make a path for the pollen nuclei. From this pollen tube, the pollen nuclei travels down to reach to the egg cell. So it is how the pollen grain reaches to the egg cell. Now what happens after this? How the fertilization occurs? So now pollen grain has reached to the outside of the ovule, through pollen tube. Now how it will enter the ovule and fuses with the egg cell? If we look at the structure of the ovule, it has outer coverings. This is the outer covering of the ovule, and these are called integuments. Integuments are for the production of the egg cell. Now, these integuments are disjoint at one point called micropyle. A small hole in the integuments is called micropyle. So the pollen tube enter the ovule through this micropyle. Otherwise, all the other spaces are closed. There is no entry point for the pollen tube. Pollen tube enters the ovule through the micropyle and it reaches to the egg cell, where it releases its pollen nuclei, and pollen nuclei and egg nuclei fuse to complete the process of fertilization. So what is important to remember here is micropyle. Micropyle is the region from where the pollen tube enters in the ovule to complete the process of fertilization. Pollen tube enters ovule through micropyle, pollen nucleus and egg nucleus fuses to complete fertilization. So this is how the process of fertilization occurs in plants. Next, let's suppose a plant has ovary, has multiple ovules and multiple pollen grains landed on the stigma of the carpel. So how will the fertilization will occur? This is egg cell, this is egg cell. Suppose it has 8 egg cells. Then 8 pollen grains will form 8 pollen tubes to fertilize the individual egg cell or egg nucleus. One pollen grain or nucleus fertilize only one ovule or egg cell. If there are many ovules in the plant, many pollen grains will fertilize many egg cells and many pollen tubes will form for this process to occur. Next is what happens after fertilization. What is the fate of the ovule, ovary and the other basic structure of flower, that is sepal, petal, stamens. So when the process of fertilization gets complete, the role of petal, sepal and stamen completes and they fall off or wither. Next is what happens to ovule. Ovule has zygote which divides many times by the process of mitosis to develop into an embryo. This we have discussed. Now the ovule will transform into a seed after fertilization and ovary grows and develops into a fruit. These two things are most important, that ovule transforms into seed and ovary develops into a fruit after fertilization. If you look at the diagram of a seed, this is a seed diagram. Seed has embryo in it and seed is the transformed form of ovule. This is the embryo inside the seed and this embryo will form in future the new individual plant and it is called seed germination. When the seed germinates, the new plant forms from the embryo. Now let's look at the structure of the seed. So we just discussed that ovule transforms into a seed after fertilization. After fertilization, it is important to write here. And the seed has embryo in it. This is the embryo, complete green one. And embryo has two parts, radicle and plumule. This is the below portion is radicle and the upper portion is plumule. Plumule will form shoot when it grows and radicle will form root portion of the future plant. Radicle will develop into root and plumule will develop into a shoot. It is written here also. Next is micropyle. Micropyle we just discussed. It is a point from where the pollen tube enters the ovule and it appears as a woolen tester. Other important feature of the micropyle is it acts as a point to take up or absorb water. It acts as an absorption point for water during seed germination. So micropyle has two important features, entry of ovule and absorption of water. Next is testa. What is testa? Testa is protective covering. This brown one is testa, that is protective covering outside the seed. And this testa is the transformed form of integuments. When the fertilization occurs and the zygote starts dividing into an embryo, the integuments or the coverings of the ovule become hard and dry. Hard and dry to form a seed coat which is called testa. Testa is important for the protection of seed. Next is hilum. What is hilum in the seed? Hilum is a scar-like region near the micropyle. It is the point from where the seed is attached or the ovule is attached to the ovary. It appears as a scar. If you see in the gram bean or green gram bean or a kidney bean, you see a scar, deep scar like this. This is the hilum near the micropyle region. Okay. Next is cotyledons. These two are cotyledons of the seed. These can be one or two cotyledons. If there is only one cotyledon, the seed is monocotyledon. Mono means single. One cotyledon. If the seed has two cotyledon, then it is called dicotyledon. these two types of plants are there, monocotyledons with one cotyledon and dicotyledon with two cotyledons. The cotyledons store food for the developing embryo. Stores food for the embryo, and food is stored in the form of proteins, starch, and the enzymes which are required for the metabolic reactions. So these all are stored in the cotyledon. You can see here that this is single cotyledon visible in this, the yellow one. And in the next diagram, there are two cotyledons visible, the brown ones. So this is the structure of the seed. Next we will see how the germination of seed occurs. When the favorable conditions arrive, and what happens in favorable condition, what we call as a favorable condition, that is water, right amount of water, temperature, or oxygen requirements. When these are suitable for the seed will absorb water and absorb water through micropyle. The seed absorb water through micropyle and cotyledons get swell. When cotyledons get swell, they start exerting a pressure on the testa or the seed coat, and the seed coat will break. Seed coat will break, and the enzymes of the cotyledons become active. And which enzymes has the cotyledon has? To break the starch, it has amylase enzyme, and to break the proteins, it has protease enzyme. So the amylase enzyme will break starch into maltose, and the protease enzyme will break protein into amino acids. And these maltose and amino acids then diffuse in water to reach to the embryo, and embryo use them for their growth. For growth, it is important that certain enzymes or enzymatic reactions, or we can say metabolic reactions, occur in the embryo. And for these reactions, food is required, proteins are required. So all these conditions become suitable when the seed absorb water. So this is the complete process of seed germination. You can see in this diagram the germination of seed. These are the cotyledons. This is the developing embryo. Developing embryo have radicle and plumule. This radical portion is forming root, you can see here, and the plumule is forming the shoot. This portion is developing as shoot. Initially, when the plant is small, the cotyledons come above the ground to perform initial photosynthesis. And when the leaves become capable enough to perform the photosynthesis, then the cotyledons will die. So this is the complete process of seed germination. Next, we will see what are the factors that affect the process of seed germination. There are three important factors that affect it. First is water, oxygen requirement, and temperature conditions. We have just seen in the condition of water, when there is no water, when no water condition is there, the testa or the integuments will remain intact on the seed, and the seed can remain dormant for years. Dormant for years because the water is important for enzymatic reactions, or water activates enzymatic reactions in the seed. If there is no water in the seed, there is no metabolism. Also, testa protects the seed from bacterial and fungal infections because it is hard covering, it does not have any water, and it also protects the seed from the harsh environmental conditions. So without water, seed can remain dormant for years. So water is important to start the process of metabolism, and then the process of growth of the embryo. When the appropriate water condition is there, root and shoot grow and the new individual plant will form. Next is oxygen. Initially, when the seed has its testa intact, then no oxygen is entering the seed, and there are initial anaerobic or no oxygen conditions in the seed. Germination occurs or starts in this anaerobic conditions only. When cotyledons swell, testa breaks and embryo gets oxygen from the environment. After this, the new cells or new cytoplasm or division of new cells begins in the seed and embryo starts growing. So oxygen is the second factor that can affect seed germination. If there is no proper oxygen for the developing embryo, the new cytoplasm or new cell formation will get affected. Next is temperature. Ideal temperature which is considered for seed germination is between 40 to 45 degrees Celsius, because it is the best temperature for enzymatic activities to occur. Above 45, the enzymes will get denatured or they will not function properly. And below 4 degrees Celsius, they will also not function properly. So ideal temperature conditions are 40 to 45 degrees Celsius. However, it can vary from plant to plant. So that's all for today. In the next lecture, we will discuss sexual reproduction in humans. Keep learning, keep practicing. Thank you.